This episode of Photonics Hot List is brought to you by Luna Innovations, enabling the future with fiber.
Hi, Happy New Year. I'm Justine Murphy, Senior Editor of Laser Focus World. Let's take a look at some of the exciting things happening in the world of photonics. A new optogenetics-based system designed to control worms' behavior is poised to accelerate medical, biological, and pharmaceutical research and potentially boost drug development. A team from Osaka Metropolitan University in Japan developed the new system and are working specifically with C. elegans, or more commonly the roundworm, a transparent nematode. For decades, the roundworm has been used as a model organism in biological research, given its multicellular structure and nervous system. In their study, the Osaka team uses two different bistable photoreceptor proteins, or opsins. Both are G-protein-coupled receptors, which sense things like smell, taste, neurotransmitters, and hormones. Opsins isolated from mosquitoes were introduced into the roundworm's sensory cells, which prompt them to leave after sensing movement. Exposing it to white light triggered that behavior. Without it, the roundworms were still. UV-sensitive opsins found in lamprey eels were then introduced into the roundworm's motor neurons, after which it stopped moving when exposed to UV light. When exposed to green light, it moved again. The findings show it's possible to optically control the signal of G-protein-coupled receptors, which could ultimately aid the creation of new devices for treating human diseases. Offering researchers at the U.S. Department of Energy's Ames National Lab a better understanding of charge transfer properties and energy conversion of a perovskite material, a new scanning probe microscope could prompt the transformation of solar cells as we know them. The team is using the new microscope to explore methylammonium lead iodide perovskite, a material increasingly known for demonstrating exceptional optical and electrical behavior for use in photovoltaic applications. It has the potential to replace silicon in solar cells. That is, if its main challenge can be overcome. As is, this type of perovskite material exhibits degradation easily when it's exposed to heat, moisture, and other such elements. The new microscope operates in the terahertz range and also allows researchers to see defect carrier traps in perovskite material's surface, as well as degradation by tracking that defect distribution over time. This may help researchers understand the local charge transfer properties and improve the global photoenergy conversion performance of this potential solar cell material. A new microscopy technique is giving a team at Johns Hopkins University an unprecedented look at organisms in 3D while also maintaining cellular resolution in all three dimensions. And they say this is only the beginning. The team used mesoscopic oblique plane microscopy, a type of light sheet microscopy, to image zebrafish larvae and a mouse brain in a larger context. They were able to capture 3 times more resolvable image points in 3D than is possible with conventional microscopy systems. Offering a more complete view and more comprehensive information about complex biological systems, the new microscopy technique produces volumetric imaging of these 3D cellular structures, including neuron circuits and blood cells, with just a single scan. The team is working to further improve the technique's performance to allow even faster and deeper imaging in highly opaque biological tissues. They also plan to explore 4D information from brain and whole biological model systems to better understand neurodegeneration diseases such as Parkinson's and MS, as well as various cancers. And that's your Photonics Hot List. For more information about these stories and more, visit laserfocusworld.com. And don't forget to like and share and subscribe to our YouTube channel for tons of cool content. Until next time.